In this talk, we are continuing with the discussion of phylogenetic methods. Also, we are going to discuss some of the techniques for the assignation of confidence levels for a phylogenetic reconstruction. Finally, we present some examples of the successful use of phylogenetic methods to solve theoretical and practical problems in biology. One limitation of both the distance and parsimony methods is that although they may select one tree over another on the basis of some criteria, it is not possible to say how much more plausible one tree is than another. In this regard, likelihood and Bayesian methods have been designed to provide a statistical framework for phylogenetic reconstruction. Maximum likelihood and Bayesian inference methods are based on statistical models of molecular evolution applying Bayes' theory. Using the Bayes' theorem is defined the concept of conditional probability. The probability of an hypothesis given the data equals the joint probability of the hypothesis and the data divided by the probability of the data. When the concept of calculating likelihood score is applied to a phylogenetic analysis, the hypotheses are the different trees and the data are the observed character traits. To do this, an explicit model of how the character stage change in a multiple sequence alignment is needed. This is called a model of evolution. These methods are very powerful and widely used for phylogenetic tree construction because they allow for testing a variety of evolutionary hypotheses within a statistical frame. Each method used to construct phylogenetic trees has its advantages and disadvantages. Here, we discussed some of the approaches that are used to evaluate various tree construction methods and the trees they produce. When the true evolutionary history is not known, a variety of tests can be used to assess the results of a phylogenetic reconstruction method. One important question to ask in this regard is, when the method has selected the ideal trees for a dataset, how well does that tree represent all the underlying data? Thus, methods for determining how well a particular tree represents the data in hand are needed. One approach to assessing how well a tree represents all of the data is to resample the data repeatedly and reperform the phylogenetic analysis to see how often the same result is obtained from these resampled datasets. These methods are known as bootstrapping and jackknifing. How these methods work? One approach to assessing how well a tree represents all the data is to resample the data repeatedly and reperform the phylogenetic analysis to see how often the same result is obtained from these resampled datasets. In the case of bootstrapping, the characters, alignment columns, are resampled with replacements. You can delete columns, multiply, or change positions anywhere in the alignment, and then recalculate the trees with the new data and compute the fraction of the new trees that reproduce the resulting tree. This is same for the jackknifing method, but in this case, the characters, the alignment columns, are resampled without replacement. For example, here we observe that some columns of the alignment are simply deleted. Here is another example of how the bootstrapping method is carried on practice. We have an alignment of sequences of 6 organisms and the resulting tree of the relations among them. Now, on the right, we have 2 examples of the bootstrap method. In the first example, or pseudo-sample 1, we observe a duplication of the first 2 columns 3 times, the third column 2 times the fifth, and 3 times the 6th column, and we delete the last 2 columns of the line. Another transformation is carried on the other sample, and so on until we obtain around 100 to 1000 datasets with these transformations. After the resample of these datasets, the resulting trees include the statistical support of each branch according to the calculation. In this tree, a value of 100 indicates a strong support, because this means that in all possible trees, the rat and human result clustered together. The other values for the other branches indicates the support of this arrangement as percentage of the number of trees that reproduce the data after the resampling process. There are other considerations of phylogenetic reconstruction. Among these, we have the do not use very small data sets. You have to select carefully the sequence to align, not too distant and not too closer. We have to take care of the alignment for mistakes. Also, we have to take into account duplications, inversions, deletions, as well as genomic hotspots, which may give wrong signals. And finally, we must pay attention to the homoplasy phenomena, which are already being discussed in this lesson. Here we present some examples of the use of phylogenetic methods to solve some theoretical and practical problems. One of such examples is the endosymbiosis theory of the origin of eukaryotes cells from prokaryotes. This theory explains the origin of organelles such as mitochondria and chloroplast in eukaryotic cells. The theory proposed that chloroplast and mitochondria evolved from certain types of bacteria that eukaryotic cells engulf through endophagocytosis. These cells and the bacteria trapped inside them enter in a symbiotic relationship. Mitochondria and chloroplast contains their own supplies of DNA, which may be remnants of the genome the organelles had when they were independent aerobic bacteria. In this sense, the single most convincing evidence of the descent of these organelles from bacteria is the position of mitochondria and plastic DNA sequences in a phylogenetic tree of bacteria. As is clearly seen from the tree, mitochondria have sequences that clearly indicates a shared ancestor from a group of bacteria called the alpha protobacteria. This is the shared ancestor. On the other hand, the plastids have DNA sequences that indicate a common origin with the cyanobacteria. Phylogenetic methods are also used to solve some practical problems, including determine the source of the infection from HIV. In this sense, the analysis of phylogenetic relationship between virus isolates help to identify viral subpopulations according to geographic localization. This helps respond to some fundamental questions about HIV origin, evolution, and diversification. Also, HIV phylogenetic classifications are used to examine the relative roles of mutation and recombination on the molecular evolution of HIV and its adaptive response to drug therapy, which helps to guide the clinical management of HIV-infected persons with adequate antiretroviral therapies according to the types of viruses. Finally, phylogenetic analysis help to understand the Earth biodiversity, the characteristics of different groups of organisms, and the evolutionary history. Biologists estimate that there are about 5 to 100 million species of organisms living on Earth today. Evidence from morphological, biochemical, and gene sequence data suggests that all organisms on Earth are genetically related, and the genealogical relationship of living things can be represented by a vast evolutionary tree, the tree of life. The tree of life then represents the phylogeny of all organisms in Earth. It implies that different species arise from previous forms via descendant, and that all organisms, from the smallest microbe to the largest plant and vertebrates, are connected by the passage of genes along the branches of the phylogenetic tree that links all life forms. Studying the phylogenetic of organisms can help us understand and explain the similarities and differences between plants, animals, and microorganisms. The Tree of Life thus provides a rigorous framework to guide the research in all biological subdisciplines, and is therefore an ideal model for the organization of biological knowledge, as well to help in the classification of new organisms from natural sources. Here we have some web services where you can obtain phylogenetic relationships among organisms. First, we have the taxonomy database. The taxonomy database is a accurate classification and nomenclature for all the organisms in the public sequence database. This currently represents about 10% of the described species of life on the planet. Also, we have the Ribosomal Database project, who provides quality control, alignment, and annotated bacteria, archaeal, and fungal ribosomal RNA sequences. These data and tools are utilized in fields as diverse as human health, microbial ecology, environmental microbiology, nucleic acid chemistry, taxonomy, and phylogenetics. Finally, the Tree of Life web project is a collection of information about biodiversity. Its goal is to contain a page with evolutive information for every species and for each group of organisms, living or extinct. Connections between Tree of Life webpage follow phylogenetic branching patterns between groups of organisms, so you can learn about the phylogenetic and evolution as well as the characteristics of living organisms. Finally, we present some of the many phylogenetic software packages that you can use to your own research. As some examples, we have the Phylogenetic Inference Package, which is a package of program for inferring phylogenetic, and the methods that they are available in the package includes parsimony, distance matrix, and likelihood methods, including bootstrapping and consensus tree. Also, we have the PO program, phylogenetic analysis using parsimony, who also include distance matrix and minimum likelihood methods. The MEGA software, which is an integrated tool for conducting sequence alignment and inferring phylogenetic trees and testing evolutionary hypotheses. It uses distance, parsimony, and maximum likelihood methods. The T-Rex is a freely available web server dedicated to the inference, validation, and visualization of phylogenetic trees and phylogenetic networks. And the Bayesian inference methods included in the Mr. Bayes program, which is a very popular program for the estimation of phylogenetic relationship using posterior probability estimations. There are many other programs used to do a phylogenetic inference as well for the visualization of the data. You can find a comprehensive list of many of them in the last link. The practical exercise of this lesson corresponds to the construction and interpretation of a phylogenetic tree.